everyone, my name is Yash and I'm a rising junior at American Heritage School in South Florida. And today I'll be presenting on my research on a novel approach to inflammaging, where I plan to test whether natural compounds like resveratrol and carnosine can actually restore immune balance in aged cells. Now first, a little bit about me. Well, I've been involved in science research since 8th grade, and most of my research focuses on immunology and longevity. I'm also especially interested in how science and policy can work together to improve care for kids with complex or misunderstood conditions like autoimmune disorders. I currently serve in leadership roles at NHS, Sigma Xi, Science Olympiad, and the Spanish Honor Society, and I am a former co-president of NJHS. Long term, I hope to become a pediatric rheumatologist and clinician researcher. By 2035, adults over 65 will outnumber children under 18 for the first time in history. That demographic shift will have major implications for healthcare. Aging is already the leading risk factor for diseases like autoimmunity, neurodegeneration, frailty, and cancer. This project is kind of my attempt to explore one of the root causes behind this growing crisis. At the center of many age-related conditions is a process called inflammaging. Now, inflammaging is a low-grade chronic inflammation that increases with age. It's marked by elevated TNF-alpha and IL-6, which are inflammatory, and reduced IL-10 and TGF-beta-1, which are regenerative. This imbalance kind of weakens your immune system's ability to regulate itself, and this contributes to disease vulnerability and impaired healing. Now, as a part of my project, I'll actually examine how inflammaging contributes to major age-related diseases. In the brain, it can damage neurons, leading to Alzheimer's and Parkinson's. In the immune system, it can trigger autoimmune responses like lupus and rheumatoid arthritis. And it also promotes tumor growth and delays wound healing by disrupting tissue repair. Now, addressing inflammaging directly could actually help reduce the risk of or severity of multiple conditions at once. Now, the problem is that current treatments like NSAIDs and steroids offer short-term relief but come with side effects and don't really address the cellular aging process. And biologic therapies are just rather expensive and often suppress the immune system too broadly. Seeing as both of these treatments aren't the best solution long-term, this means that something needs to be done about inflammaging. Therefore, the purpose of my project is actually to find a substance that can restore regenerative cytokine expression in oxidatively aged immune cells. Now, the two substances that I'll be using in my experiment are resveratrol and carnosine. Now, resveratrol is a polyphenol found in grapes and berries, and it activates sirtuins, which help reduce oxidative stress, and it suppresses NF-κB, which is a major inflammatory pathway. In my planned experiments, I'll be using it to test whether it can reduce ROS levels and promote a more regenerative cytokine profile. Now, my other substance, carnosine, is a naturally occurring dipeptide found in muscle and brain tissue. It scavenges ROS, buffers cellular pH, and prevents harmful protein glycation. And I'll be testing whether carnosine can actually protect cells from oxidative damage and help preserve production of regenerative cytokines like IL-10 and TGF-beta-1. Now, to model the immune aging, I'll actually be using the THP1 human monocyte cells, which are commonly used to study inflammatory responses. And I'll simulate aging by exposing them to hydrogen peroxide to induce oxidative stress and senescence. Now, there will be 5 groups in total: untreated control, aged cells, which are treated with only hydrogen peroxide, then aged plus resveratrol, then aged plus carnosine, and then aged plus both the treatments in combination. Now, my independent variable will be the type of treatment used, and my dependent variables will include cytokine expression, ROS levels, and cellular senescence. I'll also include positive and negative controls to kind of validate the aging model to make sure the hydrogen peroxide is actually aging the cells properly. Now, my hypothesis is that the resveratrol and carnosine, especially in combination, will reduce oxidative stress and senescence while restoring the balance of regenerative cytokines. Now, the research question I'll actually be exploring is how effective are resveratrol and carnosine at restoring regenerative cytokine expression in oxidatively aged human immune cells. Now, to measure cytokine secretion, I'll use cytokine ELISA assays. Now, these will detect levels of TNF-alpha, IL-6, IL-10, and TGF-beta-1. And by comparing expression between groups, I'll actually be able to see whether treatment helps rebalance the immune response. Now, to measure oxidative stress, I'll actually use the DCFDA assay, which relies on a dye that fluoresces in the presence of intracellular ROS, and this will help validate that the hydrogen peroxide is successfully inducing stress, and whether the treatments can actually reduce it. And then finally, I'll assess senescence using the SA beta-gal staining, where senescent cells will actually appear blue under a microscope, and this will allow me to quantify how many cells become senescent and whether the treatments reduce those numbers. So, as for relevance, inflammaging contributes to nearly every major age-related condition, but it's rarely treated directly. If resveratrol and carnosine prove effective, they could actually lead to simple, accessible interventions that help people not just live longer, but stay healthier as they age. And this project is going to be the first step towards understanding how to target immune aging with natural compounds. Now, these are my references, and I would like to thank Dr. Calkins and Dr. Shaw at American Heritage for helping guide this design of this project, and thank you to Johns Hopkins for giving students like me the chance to present our ideas and learn from each other. I'm excited to carry out this research, and I'd be happy to answer any questions. Thank you.